Olivia Hurley, don't know how to pronounce Phragmites, australis.
Got it.
Okay. Sexual reproductive effort, potential for hybridization and recovery following dieback in the lower Mississippi River Delta, and mentored by Austin Lynn. Yeah.
Good afternoon, everybody. I'm here today to talk to you about the project I've been working on for the past year under Drs. Austin Lynn and Tracy Quirk. So to give you some background, Phragmites australis, also known as rosopane, is a very common wetland reed that is found on every continent except Antarctica. And in much of the US, it's considered to be an invasive nuisance that harms biodiversity. In the Mississippi River Delta, however, Phragmites australis provides some very important ecosystem services, such as protection against erosion, waves, and storm surges. You'll see in the picture below of me holding the plant that it has long underground rhizomes which reach into the ground and help stabilize the soil. And within the Phragmites australis species, there are three haplotypes which are found in the delta. The invasive European species, or haplotype, and the naturalized delta engulf. In 2016, a condition called reed dieback syndrome, or dieback for short, was first discovered in the Mississippi River Delta. And dieback is characterized as a retreat or disappearance of these mature reed stands, and is caused by a range of biotic and abiotic factors. One hypothesis for the cause of dieback is a parasitic scale insect. You'll see to the right a picture I took doing fieldwork of healthy Phragmites stands in the background, and in the foreground are Phragmites which have experienced dieback, and all that's left is dead stems. In a region like the Mississippi River Delta, which experiences coastal land loss at an alarming rate, researchers have been interested not only in potential causes of dieback, but also potential means of restoration for Phragmites following this condition. And although dieback was only discovered in the Mississippi River Delta in 2016, it has been occurring in European wetlands for much longer. And because of that, there are trends that have been observed about how these plants respond to dieback conditions. One trend is greater sexual reproductive effort, meaning greater pollen and seed production following dieback conditions. Phragmites typically reproduce asexually through underground rhizomes, but sexual reproduction is very important in these plants because it increases their genetic variability, which overall increases their ability to adapt and overcome stressors, which is especially important in the case of biotic stressors like the parasitic scale insect. So my research is aiming to answer three main questions. The first one, how does sexual reproductive effort in terms of pollen and seed production compare among these three haplotypes that are found in the Mississippi River Delta? Second, is there any potential for hybridization among these haplotypes? And third, is there any difference in the seed bank or the natural seed storage on the soil surface between the haplotypes found in the delta? First, we hypothesize that the European invasive type of Phragmites will have a higher sexual reproductive effort. This is based off of what is known about Phragmites out of Europe. Our second hypothesis is that Phragmites haplotypes will have a very low potential for hybridization due to their different flowering times. And finally, we hypothesize that the seed bank of these three haplotypes would be different in their density of Phragmites seedlings as well as their species richness. So for the methods of this study, we visited Phragmites sites all along the Mississippi River Delta, beginning just south of New Orleans and going all the way down to the southern tip of the delta. And at these sites, we took pollen samples, collected entire infructescences or mature seed heads, and we also determined the flowering stage of the plants at each site. With the pollen samples, I conducted a pollen viability assessment in which we dyed the pollen grains and determined the percentage of viable pollen at each site. To determine seed viability, we planted the mature seed heads of all three haplotypes into healthy and dieback soil and placed them in a growth chamber to see which haplotypes would germinate best in which soil conditions. For the seed bank assay, soil samples were collected at populations of all three haplotypes in the Mississippi River Delta, including healthy and dieback sites. Those samples were then placed in the LSU greenhouse to see which plant species would germinate from them. So, time for some results. For the pollen viability assessment, we found that there was a significant difference in viable pollen production between the haplotypes. We found that the invasive European type produced significantly more pollen than the other types. We also found that out of all of the Gulf samples that were collected, none contained any viable pollen at all. With the mature seed heads, we determined the floret production in the delta haplotype to see if there was a difference between those collected at healthy and dieback sites. We found that there was in fact a significant difference, the dieback producing many more florets than the healthy sites. However, a member of our Phragmites research team was able to confirm that none of these seeds were actually viable to germinate. This could be due to us collecting seeds at a time in annual fluctuations in which none of the seeds being produced are viable, or it could be the case that the delta haplotype just produces viable viable seeds very rarely, and we just were not able to capture that within our sample size. Nevertheless, we can see that there's at least an increase in sexual reproductive effort following dieback, even if none of these seeds are able to germinate. In the seed germination test, we found that the invasive European type was the only type of Phragmites to germinate at all. And there was also not a significant difference between those planted in healthy and dieback soil. For the hybridization portion of this study, we found that there's very low potential for hybridization among the haplotypes. You'll see in this figure I made that the invasive European type begins producing pollen and setting seeds in June going through the end of summer, and the Gulf and Delta types don't begin producing pollen or setting seeds until October going through early January. Also, Gulf and Delta types are spatially isolated, and they both have a very low sexual reproductive effort, so potential for those two to hybridize is very low. In the seed bank assay, we found that there were no seedlings of Phragmites that germinated in any of the collected samples. However, we did find that the Gulf haplotype had a larger and more diverse seed banks than the other haplotypes. In conclusion, we found that hybridization between these Phragmites haplotypes is very low in the Mississippi River Delta due to temporal isolation, spatial isolation, and a very low sexual reproductive effort between the Delta and Gulf haplotypes. We also found that there is greater potential for dispersal and recruitment by seed from the invasive European type, therefore giving it the making it the best option for Phragmites restoration through seed over the other haplotypes. I would like to thank members of my lab in the wetland plant ecology lab and Dr. Osselman and Dr. Tracy Quirk, as well as members of Dr. Rodrigo Diaz's lab for letting us use their grow chambers and helping us access field sites. I'll be taking questions if anybody has any.